Today, my topic is neuromuscular junction, structure of neuromuscular junction, and mechanism of neuromuscular transmission. As you know, our body functions are controlled by two mechanisms. One is neural mechanism, another is endocrine mechanism. Neural mechanism, it is done by the nervous system. Now this nervous system are of two types, peripheral and central nervous system. The peripheral nervous system again are of two types. One is somatic nervous system, also called voluntary nervous system, and another part is autonomic nervous system, which is again divided into sympathetic and parasympathetic. So with these systems, nervous system controls our different functions, body functions, controls different movements. For performing various functions of the body, we need muscle contractions. Without muscle contraction, we cannot do so many functions, including I am talking, I am walking. So these are all voluntarily done. So how to do this work, how to control this work? So for this purpose, we have motor nerves. These are under somatic nervous system. These motor nerves, we have motor area in our brain. So we take decision in the motor area, what functions are to be done, what I want to speak, what I am to do, whether I want to walk or I am to punch someone. So these are decided by my brain, and these are executed through some nerves. They will control, they will send impulses to the muscles. So getting this impulse, the muscle will contract and will perform its function. So we can see there should be a communication between the nerves and the muscle. So this communication is done by neuromuscular junction. Okay, you understand this part? So these neuromuscular junctions are connections between motor nerve endings, the motor nerves that are coming down or coming down from the spinal cord or from cranium. They ultimately will end into the muscle in different specific muscles. So there, these motor nerves will make a connection with the muscles. So this junction is called neuromuscular junction. So what is neuromuscular junction? It is the junction between motor nerve endings and skeletal muscle and the muscles. Purpose: to transmit the nerve impulse, action potential, to the muscle so that muscle can contract. That is the purpose. Now let us see how these are done. These motor nerves, so what is it? Action potential from nerve to transmitted, nerve is transmitted to muscle through these junctions. So action potentials means impulses, means the order that has to be carried out. So it is coming in the form of nerve impulse, in the form of action potential. So this action potential has to be transferred to the muscles. Then only muscle will contract. This is a section of spinal cord. In the spinal cord, there is A-shaped area centrally. This A-shaped area is made up of gray matter. So these gray matters contains various nuclei. These nuclei are what? These are actually cell bodies of some neurons. Their axons come out from these cell bodies through different nerve roots. So nerves come out of this area, A-shaped area, gray area, through this root. These are the anterior aspect of the spinal cord or ventral aspect of the spinal cord. So this root is called ventral root or anterior root. The nerves originating from these, here are the cell bodies, some accumulation of some nuclei, means some cells. These cells are called anterior horn cells or ventral horn cells. Similar horn cells are also there, lateral horn cells. We will later know what are the functions of these horn cells. Similarly, there are accumulation of cells in the posterior aspect. These are posterior horn cells. So there are posterior horn cells, lateral horn cells, and anterior horn cells. So the motor neuron that are coming to the muscles, these are actually originated from anterior horn cells. So nerves are actually, these nerves are motor nerves. These nerves are motor nerves. So these nerves, they are nerve terminals. See, this is the nerve, this is the nerve. A nerve bundle is having, we have seen, is having numbers of muscle fibers, means axons. Okay. So every axon is ending in a, so this is a bundle of muscle. So this bundle of muscle is having fascicles. Within this fascicle, there are numbers of fibers, muscle fibers. So this axon is actually has to supply every muscle fiber, every muscle cells. Understood? Now, we need to understand one concept, motor unit, because this is important. I will explain it just after a few minutes. So you just mark, you have to know very well what is motor unit. Okay, motor unit, actually one axon, motor nerve, it is having multiple axons. So one axon, when it comes out of the nerve root, so one axon, it will produce number of branches. Okay? Now each branch will supply one nerve fiber. It may, there may be 5 branches. So each branch will supply 5 muscles. So now this axon, along with these 5 muscle fiber together, means when I will be stimulating this nerve, all these 5 fibers will be contracted. So now all these 5 fibers and this axon, motor nerve axon together is called motor unit. Now, in my bicep muscle, there may be numbers of motor unit. If only one axon is stimulated, contraction of only 5 or 3 or 6, 7 muscles will be there. So it will produce, bicep muscle will produce less contraction. If more neurons are involved, then the force of contraction will be more. How many nerves will be stimulated. It will how many nerves will be involved in muscle contraction? It depends on nervous system. It will decide how many nerves, how many fibers, how many axons will be stimulated. So they are having their respective muscles. So all these muscles will be contracted. Okay, so you understood what is motor unit? See, there are two nerves, motor unit 1 and this is 2, and these are the muscle fibers. This is supplying, this fiber, red one, is supplying this muscle and this muscle. So when you will be stimulating this, or stimulation will coming from through this nerve, it will cause contraction of these two muscles only. Two muscle fibers only, not the others. Understood? So because this is the unit. Whereas if you stimulate this one, and this one will be contracted. When both will be stimulated, all these fibers will be stimulated. So this is the concept of motor unit. Same thing. And now we'll be studying the structure of neuromuscular junction, microscopic structure. What is there in the neuromuscular junction? In the neuromuscular junction, you will get terminal button. Then what you will get? Presynaptic membrane. Then what will you will find a gap, synaptic cleft. And you will, after this, you will find the sarcolemma, muscle membrane, sarcolemma. Not the whole sarcolemma is involved. Only part of the sarcolemma will be involved. And this part that will be involved in the transmission process, this part of what? Sarcolemma. This part of sarcolemma, which is involved in neuromuscular transmission, that part is called end plate or postsynaptic membrane or neuronal end plate. End plate or postsynaptic membrane, these are actually part of sarcolemma. And the junction is such where the, suppose this is the muscle fiber. Now where the junction will be made, whether at this end or at this end or at the central position. Always at the central position, not at the, because it is made in such a manner so that because when this part will be stimulated, the action potential will be transmitted in both directions. It will be transmitted this direction as well as to this direction. Okay? Now let us see what is terminal button. These motor nerves, these are having myelination. These are myelinated nerves. Myelin sheet is there. So when this nerve is approaching to the muscle, close to the muscle, it will lose its myelin sheet. Okay? What is the first thing? When the nerve, this motor nerves will be approaching to the muscle that it would be supplying. So close to that muscle, there will not be myelination. So removal of myelination. And when this, so there will be only axolemma or axis cylinder. This may be, since it is exposed, so this can be stimulated by external factors. So what to do? These are covered by Schwann cells, glial cells. So they are now protected by Schwann cell, glial cells. These cells will cover the whole myoneural junction. This is important point. Okay, so while you will be writing, don't forget to write this. Okay, Schwann cell. Then what will happen? After removal of this one cell, the excess cylinder now will branch into, it will make several branches. These are called nerve terminals. There may be 4, 5, sometimes maybe one only, 2. So number of branches it will produce. After branching, the end of the branch will become bulbous. Will take a structure like button. That's why we call it synaptic button, synaptic knob. It's like structure will take the shape of a knob. So we will call it synaptic knob or synaptic button. So this is one of the synaptic button. So within this button, what you will get? Numbers of vesicles. These vesicles are called synaptic vesicles. Their diameter is 50 millimicron. 30 to 50 millimicron. And what they contain? They contain a neurotransmitter. And this neurotransmitter is called acetylcholine. So, thousands of synaptic vesicles are there. And these synaptic vesicles contain a neurotransmitter, and the name of this neurotransmitter is acetylcholine. Acetylcholine. Okay? In one synaptic vesicle, how many neurotransmitter molecules you can have? Guess. 5,000 to 12,000, average is 10,000. 10,000 molecules of acetylcholine is there. So, we use one term, quantum. One quantum of acetylcholine. What is that? One vesicle containing all these neurotransmitters. Means 10,000 neurotransmitters. So when we speak about quantum, immediately you think there are how many molecules? One vesicle and 10,000 molecules. This is the concept of one quantum. This is called quantum. There is a theory called quantum theory. Anyway, this is not important for you. So quantum is there. And there also you will find mitochondria. What is the function of this mitochondria here? So we know mitochondria, wherever there is mitochondria, there is need of energy locally for performing the function. So definitely it has got some energy, some function which will require energy. Because there will be some receptors, and these receptors are ATP based, sodium potassium pump. So for that pump, we require ATP. Moreover, these mitochondria, they have got other sympathetic or synthetic function. They can produce acetylcholine. The acetylcholine that will be liberated from here, what will happen to it? It will do some function. We'll see what function. And after that, what will happen to it? This will be lysed. This will be broken down by an enzyme present in here only into choline and other compound, acetylcholine. Now this choline and acetylcholine will be again coming back into there will be endocytosis. From synaptic cleft, these products will be entering into the synaptic knob, and the mitochondria will take up, or the endoplasmic reticulum will take up, and they will again synthesize acetylcholine, and again it will be packaged. Okay, so recycling of acetyl coenzyme A is there. Okay, and this is very important. Now there is Botox treatment going on, no? Botox, botulinum toxin. So these are what we are paralyzing the release of neurotransmitter from the vesicles into the synaptic cleft. That is the function. Now the rest part, acetylcholine part, will not be performed. Will not be performed. Some wrinkles are formed because of contraction. Here some wrinkles, the women don't like these wrinkles. So they, in the beauty parlor, they give Botox injection. So now this will be relaxed. Now that see, now no contraction, so it will be relaxed. So no wrinkle. Very, very good. See, my skin. These are loosened. It can be tight, make tight. So there are some processes. So you want to make some muscle flaccid. It can be done. If you want to make some to contract it, always remain contracted form, you can do it. So these are the synaptic vesicles, and this is the presynaptic membrane. Presynaptic membrane. And this presynaptic membrane is very different from this. And this presynaptic membrane, there is an area within this presynaptic membrane, we call it active zone. What? Active zone. Also called dense bodies. Dense, remember this. Active zone, dense bodies, these are synonymous. What is their function? Dense bodies. These vesicles are to release their substances, acetylcholine, into this narrow space, synaptic cleft. How they will be released? These vesicles, they are within these synaptic knob, there are some cytoskeletons. These vesicles remain attached to these cytoskeletons. Unless they are released from the cytoskeleton, they will not be secreted. They will not be, their content will not be coming to this zone. So they are to be released from there. So there is a mechanism I will discuss. There is a mechanism. With this mechanism, they will be released from there and they will be docked in the active zone, docking. They will be docked in the, they will be transferred from there to the docking zone. Then, so this will be docked here. After docking, there will be fusion of the membrane. Fusion of this membrane with this. When they will be fused, now the fused part will go apart. Now the vesicle is open. The content will come out. This is called exocytosis. So now what is happening? The vesicles are not having any neurotransmitter. So what will happen to this vesicle now? It will wait. It will wait. I release this, again I have to take up this because I know they will be broken up. So again they will be endocytosed and they will be taken back for the reformation of acetylcholine. There are several proteins necessary for removal of these vesicles from the cytoskeleton, they are transferred to this site, active site, they are docking into the active site, and their fusion in the fusion here, and their exocytosis. What are these proteins? These proteins are called snares. S-N-A-R-E, multiple snare protein. These are called snares, in plural snares. S-N-A-R-E-S. What are these? There are several, as I said, several proteins. One is synapsin. This synapsin actually binds the vesicles with the cytoskeleton. So I have to break this bond. You have to displace the synapsin. Then only the vesicles will be freed. Now it can be moved from there into the active site, active zone, into the dense bodies. And for docking, there are other proteins. And for fusion, there are other proteins. And for exocytosis, there are other proteins. What are these proteins? I will explain. So understood these points? Fine. So presynaptic membrane. So this part is presynaptic membrane, and it is having a active zone. What is the function of this active zone? Docking of the synaptic vesicles, their fusion, their exocytosis. Release of neurotransmitter into the cleft. So let us discuss the next part. Next part is what? Synaptic cleft. So before I discuss this part, sarcolemma, sarcoplasm, and within these jet bands, and these are the actins, and within the actin there are thick filaments, myosins. So these are like this, sarcolemma is like this. So when this sarcolemma is going to produce a junction, what will happen? A groove will be formed. Within this groove, what will be there? Synaptic knob. This is what? Sarcolemma. Now we will find under electron microscope, these are not like this. These are like this. Increasing the surface area of the sarcolemma. This space is called synaptic cleft. Secondary cleft is also formed here. These are called secondary cleft, and that one is the primary cleft or neuronal cleft. We also call it together motor end plate, synaptic gutter, G-U-T-T-E-R, or motor end plate. Now you understand what is motor end plate? As I said, this part will be covered by Schwann cell. So within this there are vesicles. This is the active zone. In the cleft, there will be a membrane, basement membrane, basal lamina. And this basal lamina, important. This basal lamina contains an enzyme, not various enzymes, not various, different, only one type of numbers of enzymes. What are these? Acetylcholine esterase. What is the name of this enzyme? Acetylcholine esterase. What is their function? Definitely one function. The released acetylcholine, what will happen to it after their release? If it remains bound to the receptor present in the postsynaptic membrane in the end plate, then the muscle will remain always in the contracted form unless it is released. Okay? So what to do? Are isko khatam karo. After its action will take only 1 millisecond. So immediately after 1 millisecond, sabko break down karo into choline and acetyl-CoA. So their function is this. Now, if this enzyme is absent, what will happen? The muscle will not be relaxed. So this is, in the next class I will discuss about this. These are the applied aspect. So let us now study the mechanism. So in the synaptic cleft, what we will get? Basal lamina. The basal lamina will contain an enzyme. What are these? Acetylcholine esterase. And this cleft is filled up with a fluid. These are extracellular fluid, ECF. So in the synaptic cleft, its diameter, thickness is 50 to 100 millimicron. 50 to 100 millimicron. This part of postsynaptic membrane will have number of receptors, special receptors. These are ligand-gated receptor. Here, what is the ligand? Acetylcholine is the ligand. So, these receptors are specific for acetylcholine. So, acetylcholine, when acetylcholine comes close to it, it immediately binds to these receptors and it will form a complex. When this complex is formed, this at the center, actually, what is there? Suppose this is the receptor, my finger. How many units are there? How many fingers are there? 5, isn't it? So consider it 2 alpha. Among them there are 2 alpha, 1 beta, 1 gamma and 1 delta. So these receptors are actually made up of 5 polymers. 2 alpha, 1 beta, 1 gamma and 1 delta. So when with this acetylcholine binds, with whom? One acetylcholine will bind with 1 alpha, another acetylcholine will another alpha of the same receptor. When both are become bound to this, what will happen? They change their shape. there is conformational change. There is conformational change within the receptor. As a result, a channel is opened. Because of this conformational change, a channel is opened. This channel is for sodium. And this sodium channel is again voltage gated. When a particular voltage is attained, then the diameter is increased more. So that more and more sodium ion can enter within the sarcoplasm. As a result, what will happen? If sodium ion is like other excitable cells, inside of the sarcolemma is negatively charged, outside is positively charged. So if sodium enters within the sarcoplasm, the membrane, there will be reversal of potential, means the muscle membrane, sarcolemma will be depolarized. And when this depolarization is going on, there will be reversal of potential, means there will be generation of action potential. Now this action potential will be, so here action potential will be generated and this action potential will be transmitted in both the directions. While traveling through these, there are sarcotubular system. This sarcotubular system is having cistern. Within this cistern, what is there? There is calcium ion. So when this nerve impulse will come in here, those calcium ion will be released into the sarcoplasm. Soon, initially there is no calcium ion within the sarcoplasm. In the presence of the sarcoplasm, in the presence of the calcium ion in the sarcoplasm, there will be change. There will be some sort of changes within the muscle fiber. What is it? EC coupling, excitation, contraction coupling. And because of excitation, contraction coupling, there will be what? There will be formation of cross bridges between actin and myosin. As a result of this, the Z band will be coming closer, actin myosin will be contracted, and there will be shortening of muscles. Okay? So let us see how these are the structures. Now let us see how the nerve impulse passes across this junction. What is the mechanism? Axon terminals. Can you understand this? Very clear? Synaptic knob. In the postsynaptic membrane, these are what? Ligand-gated channels, acetylcholine channels. These are synaptic vesicles, collaterals, branches, and these are the synaptic. So it is very simple. Everything is discussed. Synaptic vesicle size, where they are formed, about how many vesicles you can get in a synaptic knob. How many? This number are so many synaptic vesicles are there in a particular synaptic knob. Quantum, I gave you the idea of quantum. What is there? Other thing present in synaptic knob, already told you. Now see the mechanism. Nerve, axon. Outside, positively charged, inside negatively charged. Similar thing here. Here also positive charge, negative charge. I put stimulation. Because of stimulation, where sodium ion, where it is higher, outside or inside? Outside. So when I make it stimulated, the sodium channels will be opening. Since their concentration is higher at the outside, so there will be influx of sodium ion. So sodium ion is entering with its positive charge inside. So lot of sodium ion is entering because sodium channel is now opened because of the stimulation. Lot of sodium ion will be entering. As a result, inside will becoming positive. Outside will become negative. We call it depolarization. When depolarization exceeds threshold value, then action potential will be developed. So at this point, what will be there? Formation of action potential. Now see what is happening. There will be, inside will become positive, outside will become negative. And because of this, there will be flow of current from here to here, from here to here, from here to here, and from here to here. So see, one loop is formed. Loop is formed. As a, because of this form, this is called, current sink. So this area became current sink. Means because of this, now this current will flow on both sides. As a result, if positive charge flow from here to here, this area will become positive and this area will become negative. And this area will again become negative and this area will become positive. So now this, what happened, because of this current flow to the adjacent side, action potential is shifted from here to here, from here to here. And this area, original area, will be repolarized because of the pump. Sodium ion, they will be taken back by the pump. So this area will be in the resting state, repolarized. So because of this, with this loop formation, current will flow from here to here, here to here. Again from here, here to here. So in this way, in both directions, it will be proceeding. Clear? And the mechanism for saltatory conduction means node to node, same thing will be happening, only distance of the loop will be wider. So as a result, conduction will be higher. Mechanism is this, same, but here it is faster in case of myelinated nerve. Why? Because number of sodium channel is much higher in the node of Ranvier. So depolarization, development of action potential is faster. So as a result, their movement will also be faster. Okay, now if it go on, at its end, what is there? Action terminal. Ultimately, it will reach to the presynaptic membrane. This action potential will be arriving at presynaptic membrane. Now let's see what happens when it reaches to presynaptic membrane. So the wave of action potential through the loop formation is coming. When it arrived here, when this action potential arrived here means these are the active zones. And in the active zones, there are a lot of calcium channels. Now, because of these action potential in the active zones, arrival of active, these calcium channels will be opened. Now, from the cleft, this is the cleft. So, this area is the cleft. So, when this action potential will be coming here, so here calcium channels will be opened. As a result, what will happen? There will be entry of sodium, calcium ion. This calcium ion will activate. It will combine with one protein, calmodulin. Now this calcium and calmodulin, CALMODULIN. This calcium why calcium will be entering? Because the channels will be opened up by the action potential coming there, nerve action potential coming there. So this will cause opening of the calcium channel. Now as a result, calcium will enter into the sarcoplasm. It will bind, not sarcoplasm, sorry, in the neuroplasm. Here it is exoplasm. So it will bind with the calmodulin. A complex will be formed, calcium calmodulin complex. It will activate one enzyme, phosphokinase 2. Phosphokinase 2. Now this phosphokinase 2 will activate synapsin. And this synapsin actually binds the vesicles with the cytoskeleton. So now this will be displaced. As a result, what will happen? The vesicle will be released from there. And there are some other proteins, synaptophysin, synaptobrevin, synaptotagmin, and these proteins, these sizes for docking. With this, they will be docked here, placed there. So, for placing, these are needed. These proteins are needed. So, when calcium ion enters here, calcium calmodulin is formed. They will displace this. As a result, they will be displaced and they will be moved from there to the active sites, active zone. And by other proteins, the necessary proteins, docking proteins, they will be docked in the active site. They will be fused. The membrane will be fused, and show the picture. They will be fused, and there will be exocytosis. See, vesicle. These are the snare proteins. With these snare proteins, with the help of these synaptobrevin, these are docked. Here it is docked. See, here it is docked. Now what is happened? The membrane of these and these are fused. As a result, the contents are released. Now these are vacant. And the hydrolyzed product of acetylcholine again will enter here. And the vesicle will close and it will be used for forming neurotransmitter vesicles. Electron microscopic structure of the, so see the gutters. See the now, what is happening actually in the neuromuscular junctions. A nerve impulse is coming through motor nerve. Ultimately, it will arrive where? In the presynaptic membrane. So, arrival of the action potential, nerve action potential, where? In the presynaptic membrane. In the presynaptic membrane, what is there? Calcium channels. Now they will cause opening of calcium channel. So now the calcium channel will open. Calcium will enter from synaptic cleft into the exoplasm and it will form calcium carmodulin complex. Now this will activate one enzyme. This enzyme will dissociate synapsin from the cytoskeleton. Now the vesicles will be released. And by other proteins, snare proteins, they will be shifted to active site for docking. So another protein, that synaptobravin, it will help in docking process and fusion process and their exocytosis. So in this way, exocytosis of acetylcholine is made. Then what will happen? Now this acetylcholine will bind with the acetylcholine receptors present in the postsynaptic membrane and it will open the sodium channels. Now sodium ion from extracellular fluid will enter into the cytoplasm and the area will be depolarized. When the depolarization is coming to a threshold level, then action potential will be formed there. Now this action potential will be traveling along the sarcolemma. But what will happen to the acetylcholine? This acetylcholine cannot retain there more than 1 millisecond. Why? This will be lysed by acetylcholine esterase. Okay, now this broken product will be exocytosed, endocytosed into the vesicle. Now this will be recycled. So now what will happen in here? What will happen here? So this binding of acetylcholine with there are receptors. There are receptors here. So binding of acetylcholine with this, there will be what? Lot of sodium ion will enter within this. As a result, this membrane will also again will be depolarized, and when depolarization attains threshold value, then action potential will be developed. Now this action potential will be passing this direction. And whenever it will get the sarcotubular system, it will enter within the muscle cells and it will cause release of calcium ion. And this calcium ion will cause EC coupling. Now EC coupling will cause cross bridge formation among actin and myosin. So there will be shortening of muscles. So in this way, neuromuscular through nerve junction, through neuromuscular junction, nerve impulse is transmitted from nerve to the muscles. This is the mechanism. You understood? Receptors? Oh, one very important thing I didn't discuss. Steps. You remember these steps, mechanism. What are the incidents taking place one after another? So these are the incidences taking place. I didn't discuss two things. Next day I will discuss it. These are important for MCQs, short questions. These are what? Write down. EPP, Endplate Potential. EPP, Endplate Potential. Another potential is there that is called Miniature Endplate Potential. MEPP. Okay? Now I will take another 3 minutes. EPP. Suppose this is the end plate. This is what? Receptor, acetylcholine receptor. So here ACS will bind. As a result, what will happen? Sodium channel will be opened within this. So, lot of sodium ion will be entering. Lot of sodium ion will be entering. The inside is negatively charged, no? Like other cells, inside is negatively charged. Here, initially it was negatively charged. Now, because of entry of sodium ion, inside will gradually become positively charged. There will be gradual depolarization. Resting membrane potential of muscle is minus 80 millivolt. So what will happen? If sodium ion enters, go on entering to minus 80, minus 75, minus 70, minus 65, minus 60, minus 55, minus 50. When it will come to minus 50, all the voltage-gated channels, voltage-gated sodium channels present here, they will open. Initially it was acetylcholine stimulated sodium channel, ligand-gated sodium channel. Now when the voltage across the membrane came down to threshold level, there were a lot of voltage-gated channel opened up. So again there will be rush of sodium ion insert. So rapidly there will be change in voltage across the membrane, means there will be generation of action potential. Okay, now this action potential will be transmitted along the sarcolemma. Now question is, what is EPP? EPP, end plate potential. These potentials change where it is happening in the end plate. This is the end plate. So potential is developing here. Suppose number of sodium, calcium, acetylcholine is less, secreted less amount of acetylcholine will be secreted. So with this less amount, so less depolarization will be there, less potential will be developed, not up to the level of threshold. So this is called end plate potential. When this end plate potential exceeds the threshold potential, then only action potential develops. Understood? So EPP is the graded potential since beginning, and since the entry of started sodium ion, entry of sodium ion started, voltage across the membrane is also changing, and changing just below threshold level. This change is graded, gradually graded, and this graded change is called, in potential is called end plate potential. When this end plate potential exceeds threshold value, then only action potential will be developed and which will be transmitted along the sarcolemma. Now, the final thing, that is miniature end plate potential. It is what? Simple. Well, during resting condition, no stimulation, automatic, there are some amount of acetylcholine will be liberated from the synaptic basal ganglions under normal condition, resting condition, but very slow amount, very less amount. So this less amount of acetylcholine will bind with the acetylcholine receptors. So some sodium channels will be opened, some sodium ion will be entering, and they will produce some voltage change across the membrane. This voltage change is very minimum, 5 to 7 millivolt. So this net change is very less, and this very less change in action potential because of slow release of acetylcholine, slow entry of sodium ion, slow development of depolarization. This is called miniature end plate potential. So you will find miniature end plate potential during resting condition, not because of stimulation. And when their nerve will be stimulated, then only you will get EPP. When the stimulation is much stronger, then it will be able to produce changes beyond threshold level, then action potential will be developed and muscle will be contracted. Is that clear to you now?